So, yesterday when I recorded the lecture, it wasn't recorded, unfortunately. That's why I couldn't send. But today I'll make sure that it's recorded because many people will have to watch it. All right. So, I sent the presentation. So make sure you have calculator and you have pen and paper, right? Because today we might be doing some calculations. I hope you'll have fun because I did. So in fact, today we had to cover a lot more, but I couldn't prepare the presentation till the end, one. And two, I guess maybe it would be way more overloaded for you guys. So maybe I'll split today's lecture in two. So today it will be the first part. So tomorrow it will be the second part. And with chemical treatment, there isn't a lot to discuss about chemical treatment, so we will just discuss biological treatment in two parts. So today is the first one. Okay, so today's objective is to learn about the goal of biological treatment and to be able to design aeration tank, the tank where all the magic stuff happens. So basically by design, I mean just to be able to find its volume. And second is calculate the return sludge. I'll explain what return sludge is. So be ready for calculations today. Okay, so the biological treatment starts right after the primary treatment. And when our water is free from sand, stone, any big chunks of trash, and most of the BOD, big chunks of human waste, then the relatively purified water will go to the biological treatment. So, and the main purpose of biological treatment is basically to decrease BOD and COD. I didn't write it here, to decrease nitrogen and phosphor of the system. So it is pretty much the main purpose of biological treatment. And in Uzbekistan, so in this scheme, you see that there is anaerobic tank as well. But in Uzbekistan, I haven't heard that any wastewater treatment has one. So in Uzbekistan, there isn't one, at least up to my knowledge. And there's only aerobic part and also final clarifier. Why? Because basically in biological treatment, how the treatment is done is here in this clarifier, you have a lot of different types of bacteria. And those bacteria start eating the organic matter, and in that way they clean the water. Basically, we clean the water with the help of bacteria. And the bacteria, in fact, is in the form of sludge. They are slimy, gooey, and they look like algae, something like this. And basically, there is water, and there is this bacteria mass inside. And after the water is cleaned, bacteria still stays there. And in order to get the water separated from the bacteria, you need the final clarifier here. So everything, bacteria plus water, is treated, and then the treated water goes to the final clarifier, and then the final clarifier separates the clean water from bacteria, sends the bacteria back to the aerobic tank, and the water will travel further. So this biological treatment in fact takes a lot of time. And in Salar wastewater treatment plant, for example, it lasts from all the way from 4 to 5 days. So once water enters, it will travel. So there are a lot of parts in biological treatment. So it will travel like 4 or 5 days, and only after that it's basically treated. Until that, it's not treated till the end. So yes, now let's see more. So for bacteria to purify the water from any organic matter, the most important thing it needs is oxygen, right? So as we need oxygen in order to oxidize our food and get energy from it. Bacteria also needs oxygen. And basically, we don't give pure oxygen to bacteria. We just pump air. It's like aquarium. aquifer. I don't know in English translation. So it's like aquarium. We just pump the air inside of the biological treatment aeration tank, aerobic. So it's called aeration tank. We pump the air inside, and then the water gets saturated with oxygen and with the rest of the gases, and the oxygen is used by bacteria. Yes, so it's very necessary for bacteria to function, and this oxygen supply, which uses a lot of pumping. So now you know now why oxygen is needed. And now, for your imagination, yes, I will send some more videos of real biological treatment thing that we want, so you can. So it's a short video, and I think maybe pretty much all of you have finished watching it. And now I see more people. Okay, Alpamış, hi. And Big Zotakia, Ahmad. I hope you're present, and hello to you as well. Make your camera on. Ahmad Khan, you too, please. All right. So as you have seen here, there is bubble under the water that are pumping air, and you see that there is a huge building in the background. So this is a pumping station where they pump air, and there is a separate video that shows how this place looks like inside. So you can watch this one as well. It's also short.
Air comes from here to the aeration tank.
Yes, so these blue tubes that you're seeing in the video are basically pumping air from outside and just sending air to the biological treatment part. So this is basically what they're doing, and it costs a lot. And you hear there is like a huge background noise because pumps are noisy. Yes. So basically, this is how it looks like in Salar wastewater treatment plant. Yes. So back to our presentation. Yes. So it's not how Salar looks like when it's empty. It's a picture of some other biological treatment part from other plant. So, but the holes might look like this if they're like advanced, but I think in Salar they're not that way advanced, and they're not as frequent as here. So yes, but this is one of the examples of how this might look like. Yeah. Okay, so now we come to the design part. Whew, we should take a breath, take a moment, at least me. All right. So when designing aerobic tank right now at this stage, when we're not like neither you nor me, we're not super professional ones. The only thing we want is just to be able to find the volume of this whole thing, right? We just want to be able to find the volume. And so there are several things that you should be able to know before finding the volume. First is sludge loading rate. So sludge loading rate is explained by this Bx, and Bx has its own formula that you can see here. So it's S times Q over volume times X. So what S? S is basically the waste, the organic waste that is coming to the aeration tank, and it's expressed as BOD. The Q is the flow rate of the wastewater. The V is the volume of the aeration tank that we want to find, and X is sludge concentration. The sludge is our bacteria. So X is the concentration of the bacteria inside of the aeration tank. And usually it should be from 3 to 5 grams per liter. And so this sludge loading rate, for you to be able to feel what is sludge loading rate, is just how much we feed our bacteria. If sludge loading rate is high, it means we're giving a lot of food to our bacteria. If sludge loading rate is low, it means our bacteria is fasting. We're not giving as much food as our bacteria needs. So this is pretty much what this number means. And depending on this number, there are different types of reactors. First is highly loaded. So if this number is all the way from 1 to 2, then it means our reactor is highly loaded. From 0.4 to 1, conventional, meaning medium, average. Lowly loaded from 0.1 to 0.4. And oxidation ditch, meaning like everything gets oxidized there, is from 0.05 to 0.1. So what we want is usually 0.4. So it's like the minimum part of the conventional. 0.4, this is the number that describes how much food we give to our bacteria. Yes. And in fact, it's very important that our load stays there where we want it to stay. So why? Because if the sludge loading rate will go higher, meaning this Bx increases, so pay attention to this part, what happens is that when we give a lot of food to our bacteria, in response, our bacteria will grow larger in quantity. And when we have a lot of bacteria inside of the tank, and we don't want a lot of bacteria inside of the tank, what we do, we just recycle or throw it away. We don't literally throw it away, but we just remove a lot of bacteria from our aeration tank. Meaning, so let's say bacteria is created, like the parent bacteria divided into 2 and the child bacteria has been created. And in conventional aeration tank, the child bacteria will live like 5 days, 6, 7 days until we will throw it away. But if there are a lot of bacteria in our aeration tank, we will throw that bacteria away like after 1 or 2 days. And it's bad. Why? Because later we will learn how nitrogen and phosphorus should be removed from wastewater. And for nitrogen to be removed, this bacteria inside of the aeration tank should be at least 3 days old. Meaning, after 3 days, special type of bacteria that can lower the amount of nitrogen will start to be produced inside of the aeration tank. And once it's produced, the nitrogen will go down, right? So, and that's why we should give precise amount of food to our bacteria for it to stay longer inside of the aeration tank. So that specific type of bacteria that degrades nitrogen into nitrogen oxide or pure nitrogen that goes into the air and leaves the water. So that this bacteria would be created, right? So that's why we should try to keep this load at somewhere around 0.4. Yes. So it's a lot of information, and if you feel like you're lost, you can always go to the textbook, read it from there. And there are actually videos that explain way better than I do, right? So you can watch those too. Later I will even share. So now that we know this biological load, this Bx, so what do we do if we want to find the volume? So to design aerobic tank, which means find its volume, we just first need to select our Bx, meaning we should select whether we want 1 or 2, 0.4 or 1, or something from there. Or one thing that I should mention is, depending on the biological load, the efficiency of our aerobic tank will depend on this biological load. So you see that if our biological load If our sludge loading rate is everywhere from 0.4 to 1, then effluent BOD will be 15 to 40 milligrams per liter, right? So depending on that, you can also decide how much BOD you want to send to your aerobic tank. So what we want is 0.4, right? So that's why first we should select our BX, which is 0.4. And second, we should select our X. And X, if you remember, is just concentration of bacteria inside of the aeration tank. And rule of thumb, so again, there are no precise mathematical formulas that can describe it, or there are, but we should be a master level student. First, we should cover the whole bachelor course of wastewater treatment plant, and then we should go to wastewater. Then we should go to masters in order to understand those math formulas. For now, we cannot do so. So rule of thumb. So the rule of thumb is that the X should be everywhere from 3 to 5 grams per liter, and which means we just choose the average, which is 4, right? So now BX 0.4, X is 4, and the formula was, in order to find BX, we use this formula. So we just rewrite it, and now our formula is, in order to find the volume, we just have this C times X over BX times X, a lot of X's, right? So now we solve the problem. So here you can see that we have aeration tank, and our S is 250 milligrams per liter. Our Q is 10,000 meters cubed per day. Our BX that we have already chosen is 0.4. Our X is 4. And now I gave you like maybe 3 minutes, right? In order to solve this one. You can get it from the whatever I shared with you, from lecture slides. If you cannot access lecture slides, I am also sharing the okay. So what Sevinch sent is correct. Ahmad Khan, yours should be multiplied by 10,000, I guess. Alpamish no, Kuliora no. So the only problem here, the trick is here, is to make sure the dimensions are fine. Dimensions are okay. So you need to find volume in meters per cube, and you see the dimensions here are like in everything, like very different, right? So you should be careful about dimensions. And once you put all of the dimensions into right order, so from milligrams per liter, you will go to kilograms per meter cube, and so flow rate is okay, and X, it's 4 grams per liter. You should go to 4 kilograms per meter cube, and when you make everything correct, then you should be able to get something like Sevinch got. Yes. So dimensions. Be careful on dimensions. So I remember I was once, as I was watching some Stanford lectures on YouTube on chemical engineering, and the professor was describing that one of the workers of NASA ruined the whole space project because they were not careful about the units. So we should be careful about units, otherwise we might end up with very huge or very small.
It was miles and kilometers. Like two different groups worked on the project. One used kilometers, other one used miles.
Yeah.
Like that's why it failed.
Yes, so that's why we should be careful on that. It was pain for me my freshman and my sophomore year. All right, great. So now you can find the volume. Now the job is done. Now we move to the next topic, right? Okay. So, as I said in the beginning of the lecture, once our water is purified with different bacteria, and then what we have pure water plus bacteria, and when you look at it, it's like brownie mixture that looks no way around pure, but it's pure. it's just a lot of bacteria inside. So what do we have to do? We should put another clarifier. We looked at clarifiers yesterday, if you remember, and some of you should send me homeworks on dimensioning these clarifiers. So we looked at these clarifiers yesterday, and the basic purpose of clarifiers is to separate liquid from the solid, right? So liquid in our case is pure water, like pure water, and the solid is the bacteria, the sludge. Sludge is bacteria. Bacteria is sludge. So, and what happens is that we separate here the water from the sludge, and then the sludge goes back to the aeration tank. Or if we have too much of sludge, we will cover it tomorrow. If we have too much of sludge, we just dispose of it. We just send it to the part where a real human waste is being digested. So the sludge plus real human waste will be digested further. This is tomorrow. So the sludge that already made its job, it's sent either back to the aeration tank or to the digestion with human waste. Right? So now our second task is to be able to calculate what will be the back flow rate and what will be the concentration of the sludge inside of the back flow rate. So our assumption, so when we are doing such problem, we have several assumptions. The first assumption is that, so it's not very assumption, in fact, it should be true, right? It has to be true. Whatever comes in, the same thing goes out. So we have QI, and the same QI should leave. If we are sending 10,000 meters cube per day, then we should get those 10,000 meters cube per day back. So QI here equals QI there. And then our second assumption is the water that is leaving the secondary clarifier doesn't have any left sludge in it. So this is our second assumption. This is assumption because water might catch some of this sludge, but we just assume that there should be none. And in that fashion, we end up with this simplified formula, which means that QR, the flow rate of the water back into the aeration tank, times the concentration of bacteria in this yellow region, equals the Q. Q is just QI plus QR times XAT. X is the concentration of bacteria in aeration tank. So this is basically the formula. And in real life scenario, our first task and only task is to find this QR. But in order to know this QR, we should be able to know XR, meaning the concentration of sludge inside of this yellow region. So it will be definitely higher than the concentration of sludge in this region, right? But there is a lab procedure thanks to which we can find this XR. Yes, so there is like a mix-up. So first, this slide should be, if you are going through your own slides, go to the pre-last one. So here. So there is a certain procedure in order to find this XR. And again, reminding, XR is the concentration of bacteria in the return flow, right? So how do we do it? So I guess De La Frus, when you were last year in Hogwarts, we had a trip to Salar. So you should remember that, yes, they were doing actually this experiment. So they have like long tubes, and what they do, like here, they fill 1 liter of water from the biological treatment inside of these long tubes, and they wait like 30 minutes. And after those 30 minutes, the whole sludge settles. So before going into this whole thing, I want you to be able to imagine what we want to find. So here We want to find the concentration of bacteria. And what do we assume? We assume that the whole water is packed with bacteria to the maximum level. The water is packed with bacteria to the maximum level. And we want to find the concentration of bacteria when it's packed in water to the maximum level, right? And so what we are doing here, we are packing bacteria in water to the maximum level. We're just waiting 30 minutes, and the bacteria packs itself, like by the help of gravity, to the maximum level. So here it is the minimum volume and maximum concentration that bacteria can have, right? So, and what do? We just measure this volume here, and we call it recorded volume, V, and then we find the mass of the sludge. Here we find the mass of the sludge, and we find so-called SVI, which is sludge volume index. And then the sludge volume index is just the volume of this brownish part divided by the mass of the sludge. So recorded volume over the mass of the sludge, and we gain something that has units of milliliter per gram. So this is milliliter per gram. And this SVI shows the maximum packaging ability of our bacteria. And what do? It gets a little bit complex. So here, in order to find this maximum concentration of bacteria, we just take 1,200 and divide it by SVI. And then we can get this XR, which is maximum. And from where did we get this 1,200? So this 1,200 is just, so what they did is just they took it's just the maximum volume of sludge that can be in 1 liter of water, so which is basically 1,000 milliliter sludge. So basically, theoretically, the highest amount of sludge, which is only sludge, even no water, plus 20% excess, which means 1,200 milliliters per liter. And they just divide it by SVI, and in that way, you get XR. A bit complex here, so I guess what will be better, maybe if you go through textbook, right? Or else I may also repeat it if you want in any questions part. So imagine that we did this lab procedure and we were able to find this XR. And once we find this XR, we can already find this QR here. However, I'm giving you a reverse question where we don't know QR, but we want to find, but we know the backflow X, the backflow flow rate. So now that we should find QR by means XR by means of calculation. Yes, but in the real life, what you do is you find first QR with the help of lab procedure, and then using QR and then using XR, the concentration of bacteria in the flow rate, in the backflow rate, you will be able to find this QR. Yes. So, but now we're doing right the opposite. Now we just want to find the concentration of bacteria inside of this sludge. Yes. So again, the formula is just what I told you, and you have pretty much everything here. So you have QR, which is 400, XR, the one that you should find. You know Q, this is 500 meters per hour, and QR, XAT is here. So it's 4 grams per liter, and you should be able to find XR here. So you can start doing it. Yes, so basically this will be 9 grams per liter, and you were able to find it. And you can see that while in the aeration tank, the concentration is only 4 grams per liter, in the backflow, we have way higher concentration, which is already 9 grams per liter. Yes. And in the next lecture, we will learn what happens when there is a rainy season. So in rainy season, what happens is our flow rate increases. And when our flow rate increases, many things change. And we will find what happens when flow rate changes, the biomass of different bacteria increases, and what kind of effects does it have on our system, and how we should cope with that. And it will be covered in our tomorrow's lecture, and I will try to make it as compact as possible. Yes. So, but for today, it's all that we needed to learn. And for today, we have been able to learn how to find the volume of this aeration tank. We have been able to learn how to find the flow rate of backflow, and we should be able to find also XR if we know this numbers, but they can be found only with lab procedure, right? So with that, our lecture comes to an end. And we have covered the first part of biological treatment. Tomorrow we will cover the second part, and plus chemical treatment as well. So today, our De La Cruz wanted to show out her window session when she wanted to make a demonstration of biological treatment. However, we decided with her that it's best if she will make a short video of it and send by today's evening, so that you guys would be able to enjoy the video better. Yes, and with that, I think if you have any questions remaining, then we can answer. If not, then we can end the lecture here. So no questions. If there are no questions, then I think we can end the lecture here. So thank you a lot for your attention, and I'm waiting for the homework. Guliora, Sevinch, and Ahmad Khan. Make sure that you send the homework by the deadline. And thank you guys for your attention, and bye.